Good afternoon, everybody. Wow, that's really loud. I will talk softer. My name is Bob Burns. My co-chair is Megan Kudrowski. We're really happy to see you here, even on a lovely rainy Seattle day. We were charged by the CF Foundation to put together a molecular basis of persistent infection symposia. And what we're going to do is I'm going to show you that we have 4 outstanding speakers today. I'm going to quick advance. There's our disclaimer. There is our disclaimer. And the learning objectives of today is trying to understand how adaptive mutations occur in bacteria in the CF airway and that they promote persistence such as biofilm growth, aggregation, or specific mutations which are adaptive mutations that happen over time in the airway to both gram-negative, gram-positive bacteria in conjunction with everything else that's in the airway milieu. We also have to understand that even though a lot of us do monoculture, there is a massive amount of heterogeneity within all the strains, even in a single species. But now when you start to put it into a polymicrobial culture, it becomes a much more interesting fact of who's talking to who's listening, and who's responding. And I think you're going to see that in a number of the talks today, where we're starting to really listen well to what the different bacteria are doing. And because of this listening, fitness plus, fitness negative, are every mutation a good thing, every mutation a bad thing? We don't really know. So this is going to basically summarize a lot of the basic data on the organisms that a lot of us work on, which are the aerobes. But at the same time, over the last 10 years, and with the advent of better sequencing, better transcriptomics, better understanding of what's into the airway, mucin, the plugs, everything that's into the airway, we're actually seeing a large number of anaerobes. And I think a lot of times we have just ignored the anaerobes because they were hard to grow, or you took the sample and by the time you got it to culture, it was dead. So we just didn't know that they existed. So we have 4 outstanding speakers. We have Barbara, Matt, Nikki, and Paul. And we're going to go through Staph aureus to Pseudomonas, a gram-positive to gram-negative. Now Nikki's going to bring in the polymicrobial, and Paul's going to be the last speaker of the day talking about the anaerobes and other emerging cystic fibrosis pathogens that aren't some of the ones that more of us study than not. And there's the stenos and the Prevotellas and the whole host of other things that are really, really important. So why are we concerned about this, even in the age of Trikafta, ETI, and all these things? Because even over time, you can clearly see the top is Staph aureus. Over time, it's very high. It comes down over time, and Pseudomonas comes up. But we have a lot of organisms just maintaining themselves at 0 to 20% throughout the course of the patient lifetime. And even with treatment, it doesn't eradicate the organisms in the airway, they maintain. So even though there may be less interest sometimes in some of the pathogens that are occurring in what we're studying now, they're not going away. And no matter what we do, we're still going to have to study the Pseudomonas, the Staph, all these other ones, the Burks, because if we stop studying these over time, they're going to come back and we're not going to be prepared. So ETI therapy leads to significant reductions in the severity of respiratory infections, but it also decreases exacerbations, hospitalization, the need for antibiotics. They do see a significant decrease in the key CF pathogens, Pseudomonas and Staph, but it's not absolute. Recolonization, recalcitrant, bacteria that are going to be there for a long time are still going to have to be addressed, in my opinion. So culture positive and sputum density will also vary by individual. It will vary by mutation. We know that Delta F508 is the most prevalent, but there is a lot of other, when you have 2 of the same mutations or you're heterozygous for mutations, what does that do to your bacterial population? And then the last thing is long-term durability of the infection modulation is ongoing. The PROMISE studies are outstanding. But we still have to be very diligent in what we're looking at and how we're going to be prospectively helping future individuals, especially children who start now at the age of 2 who get ETI. What is their airways going to be in the future? And I think our speakers will start to address a lot of these questions And my goal here is not to talk anymore because you really want to hear the speakers. So thank you very much for joining our session, and I hope you enjoy it as much as I think I will. So.
All right, thank you, Bob, for that nice introduction. I'm going to introduce our speakers as they are welcome to come on up here. So our first speaker today is Professor Barbara Kahl. She's joining us from Münster in Germany. And Dr. Kahl's research mainly focuses on persistence of Staph aureus and colonization in the airways of people with CF. She's contributed in her group a lot of key studies looking at mechanisms of adaptation and persistence of Staph aureus in the airways, along with microevolution. And this is really why we wanted to bring her here today, to bring us up to date on what's happening in the field of Staph aureus research. And her talk is going to be on mechanisms of Staph aureus pathoadaptation in CF. So thank you, Barbara.
Okay. So first of all, thank you very much for inviting me and for giving me the opportunity to talk about this very important topic, which is my favorite topic in my research. Okay, so I have nothing to disclose, and I will talk about pathoadaptation of Staph aureus, and this is a huge topic, and for sure I can't really touch on all the different issues of Staph aureus pathoadaptation, and for example, I won't talk about Staph aureus and Pseudomonas aeruginosa adaptation together or alone, but about other important ways of adaptation. But first of all, I would like to give a short introduction to Staphylococcus aureus. This is a very important pathogen, not only for CF patients, but is important also for all other patients. It's a pathogen or a commensal which colonizes a third of all people, and persistently, and 30% intermittently, and 30% are almost never colonized. And it can cause severe and also mild infections. So superficial infections like skin lesions, and also other infections like toxinosis, soft tissue infections, device-related infections, bacteremia, sepsis, endocarditis, pneumonia, osteomyelitis. And for this talk, I only concentrate on the lungs of the CF patients, but maybe I just can touch on this. You should think of the CF patients who are persistently and often colonized and infected by Staph aureus in large percentages, but we almost never see sepsis or bacteremia, so they are somehow protected, and especially their adaptive immune system is very good in order to keep Staph aureus to the lungs, although they have tons of Staph aureus sometimes in the lungs. So this is very special to the CF population, and in intensive care unit we see this very differently. They get bacteremia and sepsis very fast. Okay, so this only is an outlier. Okay, so you just saw this graph with the Staph aureus prevalence in CF, and especially in the young patients, you see a lot of Staph aureus, and it declines at the agings, and Pseudomonas aeruginosa is coming up. But we all know that this has changed because Pseudomonas eradication is very good, and the prevalence of Pseudomonas is really going down. The prevalence of Staph aureus is going up. And here you can see the prevalence of MSSA and MRSA, which is MRSA is only a part of the entire Staph aureus prevalence, and in many studies it has been shown that MRSA especially has a worse effect on CF patients. Oops, sorry, this was the wrong one. Okay, but to give you a flavor of the persistence, what this really means, here are data from the European CF Foundation, and you can see, so these are data of chronic Staph aureus infection, and I picked some countries with their prevalence of chronic Staph aureus infection, like the UK, with 7% in children and in adults 6%. Although this morning we heard a talk from a study in the UK where the prevalence of Staph aureus was much higher, which I really don't understand. These are data from the European CF Foundation. And in Italy, it's about 30%, France 40%, in children, in adults 30%, and in Germany it's quite high, 40% in children, 46% in adults. And persistent means at least half a year positive for Staph aureus, or 50% of the specimens of 1 year positive for Staph aureus. And these are data from 56,000 patients in Europe. So many patients persistently infected or colonized by Staph aureus. Sorry, this is the wrong direction. Okay, and here I will show you data from our study in Münster, a long-term observational study, which I started in 1994, and we have data from 1994 until 2016. And these are data from 2 centers in Münster, and you see data from 200, almost 300 CF patients over these 22 years, and short description. I won't get into detail in this heat map, but if you see, you can see the years from 1994 until 2016 on the x-axis, and the individual patients on the y-axis. And if you see a red button, then it means there are 4 samples positive for Staph aureus in that year. If you see a pink button, it means there are 3 at least positive for Staph aureus that year. And if it's blue, it means there's no Staph aureus. There were cultures, but no Staph aureus in that year. And without going more into detail, you can see that many, many patients are persistently colonized, infected by Staph aureus. So these were, 2,867 patient years, and persistence was 61 patients or people with cystic fibrosis were persistently infected, meaning there are many, many patients with people with Staph aureus in their lungs. And one of the very prominent adaptation procedures that Staph aureus is using is the emergence, or the way of changing its molecular or metabolism, and it really, really looks very different. It's the small colony variant phenotype, and here you can see on this agar plate the normal Staph aureus and the very, very small ones. And if you look at these special small colony variants under the electron microscope, the small colony variant bacteria are much larger compared to the normal Staph aureus, because they are impaired in their replication. They don't replicate, they just grow and grow and grow, and the single cell is getting larger and larger. And here you can see several division planes in such a bacterium. And these small colony variants, they have a huge advantage. They can survive intracellularly much better than the normal Staph aureus because of their downregulated metabolism. They don't produce alpha toxin and other toxins, which keep them intracellularly, and the normal Staph aureus grows and destroys the cells and gets out. And by this, the bacteria, the small colony variants, are protected against antibiotic therapy and also against the immune system. And here you can see it's an old study. You can see an old Northern blot where you can see that all the virulence factors are downregulated, and also the regulators like RNA3, the AGR regulon is downregulated, and HLA alpha toxin is downregulated, and protein A is upregulated. So adhesins are upregulated and the toxins are downregulated. And these are special small colony variants which also have been isolated in the US a lot, the thymidine-dependent small colony variants. And they have a special mutation, which I will explain a little later. And if they are complemented with thymidine, then they can grow and behave like the normal Staph aureus. And there were especially from Seattle, great studies from Luke Hoffman and this group, Dan Walter, the first author, they showed that people with small colony variants in their lungs, they have worse lung function compared to people with CF with only normal S. aureus. And we also could show this a couple of years ago in another study that the people with small colony variants had worse lung function compared to the normal Staph aureus. And here are some studies collected. They were, I started in the review a couple of years ago to collect the studies, and I tried to actualize or to update them every once in a while. And there are many, many studies showing that CF patients have this small colony variants in their airways. And the most beautiful studies were done here in Seattle showing the worse lung function. So these are data from more than almost 3,000 patients from 11 different countries. So they are in many, many different countries available. And just to explain this phenotype, the small colony variants, which are thymidine dependent, they are induced and selected by trimethoprim sulfur methoxazole treatment, because the trimethoprim sulfur methoxazole interferes with the tetrahydrofolic acid pathway, which you can see here. And tetrahydrofolic acid is a cofactor for thymidylate synthase. And when the patients are treated for long term with this antibiotic, in the CF background or in the CF lung, mutations occur in thymidylate synthase, which usually is lethal for bacteria or for Staph aureus, but in the environment of the CF lungs with lots of pus and destroyed cells, they can take up, especially by this nucleotide transporter, they can NACFC, they can take up thymidine and supplement their mutation with this extracellular thymidine. And by this, they can behave like the normal Staph aureus and can survive, although they have this essential mutation or lethal mutation. And all these small colony variants, thymidine-dependent small colony variants, are resistant against trimethoprim sulfur. And Luke Hoffman, he showed years ago that another type of small colony variants can be induced by co-infection with Pseudomonas aeruginosa, especially with when substrates of the Pseudomonas aeruginosa were challenged with this Staph aureus and induced the small colony variant phenotype. And these were human-dependent small colony variants. They had problems with the electron transport chain, and they are always resistant against aminoglycosides because the membrane potential is going down and is not working, and the aminoglycosides are actively or have to be actively taken up into the cell. And another group from Germany, they showed that also pyacyanin, another very important virulence factor of Pseudomonas aeruginosa, can also induce this phenotype. But this is different compared to the story of the thymidine-dependent small colony variants. And there are lots of more small colony variants without so far known underlying mechanism. Some of them are CO2 dependent, and there are also lipid-dependent small colony variants, and more to go. Oh, sorry, this is again the wrong direction. And there was a beautiful study from Tang et al. And also Luke Hoffman was a co-author. They showed that, what they showed might explain the effect of small colony thymidine-dependent small colony variants on the lung function of the CF patients. They showed that by disruption of the thymidine synthesis, the bacteria had lots of CDAMP, the second messenger, which induced robust sting activation, and with the sting activation, this leads to a higher airway inflammation, and this might be a reason for this worse lung function in people with CF and small colony variants. The authors could not show this same mechanism for hemin or menadiene-dependent small colony variants, so this only affects the thymidine-dependent small colony variants. Okay, and another study, also again from the Seattle group, they had also a beautiful study from 5 US CF centers with more than 1,300 Staph aureus isolates, which they collected during a 2-year observation period. And they showed that there was convergent evolution in Staph aureus isolates in children. I really emphasize children with CF during this 2 years observation. And what they could show is, or what they could see, is that there were convergent evolution mutations in the rifampin resistance, gene RPOB, and tetracycline resistance, and also the thymidylate synthesis gene, the TMP-SMX resistance, and also mutations in the AGR regulant, which we also could see in the small colony variants with reduced virulence. So this was very interesting. But I think this is special for children with Staph aureus being infected, colonized for a couple of years. This might be very different for older patients who are longer persistently infected with Staph aureus. And another very interesting finding that they have had was that they could show or could see that Staph aureus, in contrast to Pseudomonas aeruginosa, has an open pangenome, meaning that there was an ongoing acquisition of new genes among the Staph aureus population, which infected the airways of CF. In Pseudomonas, it's a closed pangenome. So this might explain also the variability and flexibility of Staph aureus. Okay, and so we did some studies also showing adaptation to polymorphonucleotides, which the Staph aureus sees a lot in the lungs of the CF patients. And so Staph aureus expresses 2 superoxide dismutases, SOT-A and SOT-M, which are important to detoxify the superoxide radicals, which are produced by the neutrophils. And interestingly, this SOT-M is unique to Staph aureus, which is the more virulent bacterium in comparison to coagulous negative Staphylococci. And this SOT-M has a special function because it can survive the function of, the function of calprotectin, which is expressed or secreted by the neutrophils. This is important for the nutritional immunity of the PMNs. They scavenge manganese and zinc, and the SOT-A uses manganese, but the SOT-M can also use not only manganese, but also iron in order for its activity. And we could see in our persistent Staph aureus isolates that, so here you can see the SOT-M transcription of sequential strains from 6 patients, the early and late, so at least 5 years cultured apart. And they were clonal, which we showed before, and the SOT-M was much higher in many of the strains because such higher expressing SOT-M isolates were selected mostly due to the effect of the superoxide radicals produced by the neutrophils. And to show that this is also in vivo relevant, we we did some metatranscriptomic studies from sputa, here from three patients, 2 sputa from each patient, and these are different virulence factors that we looked at, and they were active, but much more active were the superoxide dismutases from the sputa directly isolated. And another very important way of interfering with bacteria are the neutrophil extracellular traps. These were mentioned already yesterday, and there is a nice study from the group of Maren von Köckritz-Blickwede. She showed that Staph aureus expresses a nuclease which facilitates the escape from neutrophil extracellular traps, so that Staph aureus is not killed by neutrophil extracellular traps, by nets. And so we hypothesized that something like this must also occur in our persisting strains. And we looked at, first of all, at net production in CF sputa, and you can see here the nets in a sputum of a CF patients, and also FITS labeled Staph aureus. And you can see, these are persistent strains from one patient and over 13 years, and the nuclease activity by RT-PCR, and there was much higher nuclease activity in late strains. So this is also, and also in the metatranscriptomics, nuclease activity was very active. So high activity of nuclease in sputa, highlighting the important role of nuclease in Staph aureus and the adaptive phenotype with higher expression of nuclease. So, and the last phenotype which I would like to introduce is the mucoid phenotype of Staph aureus, which we got interested a couple of years ago. And here you can see the very mucoid Staph aureus. You can see this in the movie. It's like chewing gum, and it's very difficult to see on the agar plate. You really have to touch it by the loop. And this is caused by a deletion in the intergenic region of the ICA operon, which is responsible for the production of polysaccharide-dependent biofilm. And you can see the biofilm, the huge biofilm of this phenotype in this electron microscope. And here, so this very high producing biofilm bacteria are protected against killing by neutrophils, and this really might be the mechanism why these bacteria are selected during the persistence. And we did a prospective multicenter study. I think I have to hurry up a little. A prospective multicenter study to look how prevalent these mucoid Staph aureus are. So we looked at more than 600 patients from 13 centers in Europe and one in Munich, in Austria, and we could find a prevalence of almost 10% of these mucoid phenotypes. And interestingly, there was also a high prevalence of small colony variants. And we also got in a longitudinal study data from clinical data, and we looked for 1 year at different visits if the patients are and compared them to a control group with only normal Staph aureus, if these mucoid phenotypes have an impact on lung function. And what we could see that only in patients without Pseudomonas aeruginosa and in female patients, these mucoid phenotypes were associated with adverse lung function. And we also did some more studies on the molecular background. We did not only find this 5 base pair deletion in our mucoid strains, but also deletions in the regulator of this intercellular adhesion operon, and we did a study on that. And there are still a number of molecular mechanisms which we don't know, and this is ongoing work in our group. And now we try to mimic this situation in the CF lung by trying to induce this mucoid phenotype with experimental evolution experiments, with looking for 10 and 20 days in a biofilm behavior, and trying to find this or induce this mucoid phenotypes. And we could induce it in many, many different situations with various stressors like salt, glucose, oxidative stress, anaerobic conditions, and antibiotics. And we also used reference strains like SH1000 and GA21, and in all strains we could induce this mucoid phenotype. And looking at the Ica-operon then with there was not this 5 base pair deletion, which we find for the first time, but we found in these strains, after 20 days, many different mutations in the regulator of this polysaccharide regulon. And just as an outlook, so Staph aureus is a master of adaptation to the airways of patients with people with cystic fibrosis. There's a long-term persistence of clonal isolates, partly due to SCVs, mucoid and other phenotypes. S. aureus is characterized by an open genome with influx of new genes, including virulence factors and resistance genes. And further investigations will look at the unknown mechanisms conferring the mucoidy. Is it protein related or is it due to regulation? Special role of mucoid SCVs in terms of survival and competition assays, the host pathogen interaction of mucoid normal and mucoid SCV isolates, and an impact of mucoid SCVs on lung function. And with this, I would like to thank all the people who did the work, the funding agencies, the CF centers who contribute, and especially the patients, and you for your attention. And I'm happy to take any questions. And it's 000.
Perfect. All right, there's a microphone in the middle of the room. If anybody has questions, feel free to come up. You're also welcome to enter your questions in the app, but if you're here and you want to ask them in person. Any questions?
And please identify yourself and your affiliation before asking your question.
It's not working?
Oh, might not be on.
Onzi from Italy. So thank you, Barbara. Was a very nice overview as usual. And I would like to ask whether are you expecting differences in patients treated with CFTR modulators, and if you are any update on that.
Okay, thanks. Thanks, Alessandra. So when our study was running, this prospective multi-center study we started in 2019, were affected by the pandemics and also by the ETI beginning of the treatment, and the study ended in 2022. So we already had patients being on modulator therapy, and we really, also analyzed if ETI has an impact, but we could see that many of the patients with the mucoid Staph aureus, it still could be isolated. Although I have to admit, so from our so this was shortly or at least 1 year after ETI, beginning of ETI treatment. So looking now at our clinical diagnostic lab, we still find them, but with the throat swabs that we mostly get, not sputum anymore, this might differ. So we don't find them that often anymore, but we can find them. But so in our study, we also did a more sophisticated or more elaborated culture with getting 10 isolates from one specimen and looking for the small colony variants and also for the mucoid isolates, and this also might have an effect. So it's really important to look very, very carefully.
I thought the observation that STING signaling could change SCV formation or support different SCV phenotypes was really cool. I wasn't aware of that, but so STING can be activated by antiviral signaling and other inflammatory stimuli that could change the lung environment. So I'm wondering if we know much at all about how maybe viral co-infection in CF might be driving any adaptation. And I know that there's some really nice work looking at metabolism driven by things like viral infection and stuff like that as well.
Yeah.
So I don't know if you could comment on that at all.
No, sorry. So I'm not a viral person. I didn't do any work on viral Staph aureus co-infection. No, sorry.
Okay. Other questions? So thank you, Barbara, for
Okay, thank you.
All right, I'm excited to now introduce our second speaker. So we'll move on from staph, and I have to talk about Pseudomonas. And so I'm very excited to introduce Matt Parsek, who's professor of microbiology at the University of Washington. He came a really long way to be here, all the way from north up by the lake. And so Matt has done some really nice work in his group about Pseudomonas biofilm matrix, biofilms in the host environment, and surface sensing by Pseudomonas aeruginosa. His lab does a great job of kind of combining and bridging biofilm work in the lab and also understanding how this translates to bacterial aggregates in vivo. And so he's going to talk to us today about how Pseudomonas aeruginosa persists in the airways and some mechanisms that Pseudomonas uses to adapt in the CF environment. So,
all right. Thanks, Megan and Bob, for the opportunity to share with you some of the work we've been doing in my lab. You kind of gave part of my intro, Megan. So today I'm going to start off by reviewing a bit of what we know about how Pseudomonas produces aggregates. And then I'm going to talk about some of the stuff we've been doing, trying to figure out how in vitro, what are some of the basic principles Pseudomonas aeruginosa uses to produce aggregates. And then move to the in vivo environment and ask, are any of the things we learn in the laboratory relevant to what's going on in CF? Okay. So I assume many of you are familiar with Pseudomonas aeruginosa. It's found everywhere, and it's also a very versatile, opportunistic human pathogen that's important in CF airway infections. And another reason we're interested in Pseudomonas aeruginosa, it's a model organism for studying social behaviors in bacteria, something we call sociomicrobiology. And a couple examples of sociomicrobiology are quorum sensing or intracellular signaling and the formation of biofilms. So my lab over the years has focused a lot of its energy studying the Pseudomonas aeruginosa biofilm matrix. And there's many questions we've asked over the years. What is it composed of? And in this particular little schematic, this hazy green web surrounding the individual cells that are attached to a surface represents the matrix. So we've asked, what is it composed of? And depending upon the strains, strain of Pseudomonas, we found different things. We know that exopolysaccharides are important, proteins are important, and nucleic acids. We've also asked, how is its production regulated? And it's tightly linked to the signal cyclic diGMP. And we've also studied the functions of the biofilm matrix, which are can be structural, protective, and also be involved in signaling to the bacterial community. So cyclic diGMP, like I said, is a key signal, intracellular signal that regulates matrix production. So levels of cyclic diGMP in the cell can vary. There's a number of proteins that can make it. There's also a number of proteins called phosphodiesterases that can break it down. And the basic idea is when it's low in the cell, that favors motility. And from the standpoint of Pseudomonas, it's also important for acute infection. When it gets high, this favors biofilm formation and chronic infection. And some of the factors that are upregulated under conditions of high cyclic diGMP are the exopolysaccharides PSL and PEL, as well as an extracellular biofilm matrix protein called CDRA. So let me tell you a little bit more about these matrix components. These two polysaccharides, PSL and PEL. Now, PSL is a neutral exopolysaccharide. It's a pentasaccharide subunit. It is shown here. It's neutrally charged. The other, so there's a single operon responsible for its production. There's another exopolysaccharide called PEL. PEL is a positively charged exopolysaccharide that's primarily N-acetylgalactosamine, okay? And CDRA is this adhesin that actually binds to these two exopolysaccharides and acts as a cross-linker stabilizing the biofilm matrix. Now, in the case of Pseudomonas, we know that depending upon the strain, you can sort of classify them into 4 classes based upon their exopolysaccharide dependence in forming biofilm. So if you look at the y-axis here, this is relative biofilm formation in vitro. And here are some representative strains of each class. Class 1 is PEL dominant. So basically, in the case of PA14, if you mutate PEL, you lose the ability to form biofilms. Conversely, class 2 are PSL dominant matrix producers, and PaO1 represents this. In PaO1, if you mutate PSL, you lose the ability to form biofilms. Then there are what we call EPS redundant strains, where you have to mutate both PEL and PSL to eliminate biofilm formation. And then we have matrix overproducers that produce a ton of biofilm, and it's due to overproduction of both PEL and PSL. Okay? We know that clinically we can find representatives of all 4 classes in CF isolates. The biofilm matrix protein CDRA, I'll tell you a little bit more about that. This work is linked to a former postdoc of mine who now is running his own lab at Colorado State. And it's this long filamentous adhesin that basically, under conditions of high cyclic di-GMP, is trafficked out of the cell. And here's a model of its structure. And this is the protein that interacts with the different exopolysaccharides cross-linking it. Now, an example of its importance is if you take a strain, so this WISPF mutation makes the lab strain PaO1 a hyperbiofilm former, or a class 4 strain. And under conditions where it's making a ton of matrix, you can see this WISPF strain. So what you're looking at here is the picture of an aggregate taken from a flow cell biofilm, okay? The cells are labeled in green, and we took a lectin that's specific for the exopolysaccharide PSL that's labeled with a red fluor. And you can see that aggregates, so this is a bottom view looking through the attachment surface, which is a glass slide. You can see there's an aggregate of cells surrounded by this shroud of PSL, and here's a side view. And if you mutate this adhesin, you lose the ability to tether and maintain this PSL shroud. So it's very important for structural integrity. Now, one thing that we've discovered that is really interesting about these different exopolysaccharides is that depending upon the exopolysaccharide, its localization in a biofilm aggregate can change. Now, what you're looking at here is the same aggregate. In green is all cells, so all biomass. So here's a top-down view of the aggregate. Here's a side view, okay? If you stain with the lectin specific for the exopolysaccharide PEL, we see that a lot of it's found at the surface, a little bit in the interior of the aggregate. Whereas in the case of PSL, we find that it forms this nice shell surrounding the aggregate. Now, what we found, this is basically the same data I showed you, looking at all biomass in the 2 different exopolysaccharides. Now, if you eliminate the ability of this strain to make PSL, what happens if you lose this shroud of PSL, this extracellular shell? We see that in this case, the strain compensates. So this is a PSL mutant in this background. The strain compensates by using PEL as its extracellular shell of EPS. So this led to the question, is an EPS matrix shell a common feature of microbial aggregates? All right, one thing we know that is that in vitro, as time goes by, here's a side view of a biofilm growing on a glass slide. You can see it starts as a little mound, and as time and growth occurs, it matures into a much bigger aggregate, okay? And the question is, as it grows, how does it accommodate new biomass and maintain this shroud or shell of EPS? Okay, this is going back to the localizing PSL. So here PSL is labeled in sort of a purple color. Here's a top-down view of an aggregate. Here's a side view of the aggregate. You can see that you have this region of real dense biomass with thousands of cells surrounded by this about 10 to 15 micron wide zone that doesn't have many cells but is very rich in the exopolysaccharide PSL. So we asked the question, is new matrix production required for aggregate growth? And to do this, we took a strain. So this is a PA01 strain where we got rid of PEL, mutated PEL, and now we express PSL conditionally under the arabinose-inducible promoter PBAD. So what you're seeing here is biofilm formation on a glass slide where we're supplying arabinose in the growth medium continuously as it flows through the system. And after 24 hours, you can see this strain has formed a nice aggregate. Here's a top-down view, a side view down here. Once again, PSL is stained in purple. And you can see that you have this shell of PSL that's being produced that surrounds the aggregate, very similar to what I've already shown you. Now, at this point, what we did is we either maintained arabinose in the growth medium or we removed it. And we asked the question, how does this impact continued aggregate growth? And what we saw is if we maintained arabinose in the ability to produce PSL in the growth medium, the aggregate got bigger, as you might expect. In the case where we removed arabinose at 24 hours, we saw the aggregate stayed about the same size. Very stable. We could still stain PSL in the periphery, but you couldn't continue to grow as an aggregate. So the matrix appeared to be fairly stable, but for continued growth, the loss of the ability to produce PSL was crippling for aggregative growth. We did the same strain with same experiment with PA14 using a PBAD PEL producer. Okay, so arabinose allows production of the PEL polysaccharide. And if we did the same sort of experiment, supplying arabinose in the growth medium, we got nice aggregates that formed in PA14. If we maintained the presence of arabinose, the aggregates got bigger. And like the PSL experiment, if we removed arabinose at 24 hours, the aggregates stayed the same size. Okay, so this led us to sort of propose two models that frame matrix dynamics. The first model we called the expanding balloon hypothesis. And the idea is that as biomass grows, that the EPS shell surrounding the biomass just expands to accommodate the growing biomass. So EPS, of course, is labeled here in pink. And one way to think about it, here's the balloon down below. And if you labeled different regions of this EPS shroud, as the balloon gets bigger, biomass grows, you would expect these labels to remain in the periphery and relative to one another. Another model is what we call the tree ring model, and the idea here is that as new biomass grows, that you're continually producing new EPS and recycling old EPS, sort of like layering on new biomass at the, in a tree as it grows. Okay, so one way to think about this is matrix accumulation versus matrix production. Matrix accumulation equals matrix production versus matrix loss. And a way to test these two models was we did an experiment where at a given time point we applied a lectin and allowed the biofilm to continue to grow. Okay, so the idea would be if the expanding balloon hypothesis is right, the labeled EPS should remain in the periphery. Whereas if the tree ring model is more appropriate, you would see that new EPS would be layered over the stained EPS. So what did we see? We did this experiment, and at 72 hours, we stained with a fluorescent lectin for PSL, and we monitored the same aggregate over time, and we could see the aggregate was getting bigger. And then we monitored where the labeled EPS was relative to the growing aggregate over the course of the experiment. And what we saw is that the PSL, if this is sort of the area of the cross-section of the aggregate, and over time, remember we labeled at 72 hours, so we could see that the aggregate is getting bigger, but the location of the stained PSL remained in the same spot. So this really favored the tree ring model. Another way we tried to do this, test these models, was to use fluorescent beads. Now we knew this cell, this region surrounding aggregates that didn't have many cells and were EPS rich could be labeled with beads, fluorescent beads. So we could add beads to existing biomass and image this peripheral region using a confocal microscopy. So this is the same aggregate, and you can see the beads have labeled this peripheral shell of EPS. And if we allow the same aggregate to grow and do the labeling experiment again, what we saw is here at 96 hours, we labeled with red beads. And we can see we labeled the peripheral shell. And after 144 hours, we added a different color bead, did the labeling, and saw that it was exterior to the internal labeling. Okay? This once again suggests that the tree ring model of EPS production is correct. So, sort of revisiting the two models, our data is consistent with hypothesis number 2, or model 2. Another point is that the matrix is very stable, except when the aggregate is growing. It appears that the matrix is being recycled as new EPS is being produced. So biofilms require new matrix production for aggregates to grow. New matrix is layered over the top of existing matrix, and the matrix is fairly stable. Now we ask the question, what about aggregates during CF infection? Is any of this relevant to what's really going on in disease? So we know that in Pseudomonas that people with CF get defective mucociliary clearance, okay? And it results in a buildup of thick viscous mucus that's colonized by Pseudomonas and other bugs. And we've known that this becomes colonized and you get the formation of very large aggregates of Pseudomonas in this mucus, which is in purple here. The yellow or the white arrows are indicating Pseudomonas, call them macrocolonies. You got thousands of bacteria present in sputum. So we decided to try and label both Pseudomonas and the exopolysaccharides using immunohistochemistry. The idea is that we can take clinical sputum samples, PCR amplify for both Pseudomonas and the presence of the PEL and PSL operons. We can also plate or freeze away some of our sample, and then we can use some of it for immunohistochemistry to look at the presence of both Pseudomonas and PEL and PSL. Okay, so the basic protocol is simple. We fix sputum in formalin, we embed in paraffin, we slice and mount sputum sections, and then we stain and do a immunohistochemical analysis. So we have antibodies specific to PEL, antibodies specific to PSL, and antibodies specific to Pseudomonas. So one of the first control experiments we did was to take someone with a Pseudomonas aeruginosa negative sputum sample and spiked in some lab-grown Pseudomonas and make sure that we could see it. Here's an example of an aggregate that's producing PEL that we added to this negative sample. And after a lot of optimization, we were able to do the interesting experiments to ask, does Pseudomonas form aggregates? Are PEL and PSL produced in these aggregates? And do PEL and PSL signals, if present, co-localize with Pseudomonas? And here's an example of one sample that we looked at. Here's the negative control. If we stain with Pseudomonas, we could see the presence of aggregates of Pseudomonas, which isn't a big surprise. And in this particular case, we saw that anti-PEL and anti-PSL signal co-localized with the presence of these aggregates. A bit clearer sort of picture. So here's an aggregate of Pseudomonas in green, stained with anti-Pseudomonas aeruginosa antibody. And you can see it's surrounded by this shell or shroud of PEL that's stained in brown. So this is somewhat consistent with some of the patterns we saw with our in vitro work. Here's just a negative control. These are gram-positive cocci that, luckily didn't stain with PEL or Pseudomonas antibody. And this summarizes a lot of the study. So we know that aggregates, depending upon the sputum sample, so here are 3 sputum samples compared to a flow cell grown biofilm in the laboratory. And a couple things stand out is that in general, most aggregates are small in size, so on the order of 20 to 40 microns, which are definitely smaller than the aggregates we generally see in a fully grown flow cell biofilm. We found that a lot of these aggregates had both PEL and PSL signal associated with them, but we did see cases in some patients where it was primarily either PEL or PSL as the major signal. So one question is, why should we care if these matrix components are being produced by Pseudomonas during CF infection? Okay, and our thoughts were, well, they might impact antimicrobial activity. And the point I'll address is that there's a strong potential that they can influence interactions with host-derived material. And a lot of this thinking is directed towards the exopolysaccharide PEL, which is this positively charged exopolysaccharide. And the positive charge present on the amino group of PEL would have a high potential to interact with negatively charged things in the host environment. And indeed, if we looked at Pseudomonas making PEL, so what these are cell-free extracts where we have PEL present in the cell-free supernatant. Here's a negative control that isn't making PEL or PSL, and here's a strain that's making primarily PSL. So we get rid of the cells, we have the supernatant that's rich for PEL or PSL with a negative control, and then we add CF sputum fluid. And we did this at 2 pHs, and the reason we did this is at this pH, PEL should be positively charged, and this pH is above the pKa of PEL, so it should remain neutral in the case of PEL. And indeed, what we saw is that in CF sputum, there are things that are complexing and aggregating with the PEL being produced by this strain here. We didn't see this in the PSL overexpressing strain or the negative control. Okay, we went on to show that actually DNA is a very strong candidate for interacting with PEL. And once again, these are the same sorts of experiments where we take the cell-free supernatant of strains. Here's our negative control. Here's our PEL overproducing strain, PSL overproducing strain, and a strain that's making a lot of alginate. And in this case, to these cell-free supernatants, we exogenously added DNA, so eDNA, which is rich in the CF airways. And of course, in the case of PEL, when the pH is right, so PEL is positively charged, we saw the formation of an aggregate. Now we asked the question, does this PEL eDNA aggregate influence any of the therapies that we use in CF? And one of the common therapies is exogenously applied DNase treatment to CF airways. So what we did is we did a 48-hour digestion of this complex. It's either with DNase for 48 hours or a negative control. And we could see that we still saw an aggregate after 48 hours of DNase treatment. If we looked at the presence of DNA in the non-aggregate part of the sample, so in the soluble part, we saw there was no eDNA present, even though in the negative control we could see a strong signal. However, in the aggregate, the presence of this PEL eDNA complex protected the eDNA from degradation. We could change the pH of this so that the PEL polysaccharide was no longer complexed, and the released eDNA at a higher pH actually allowed DNase to do the job. So it looks like, this complex can protect eDNA from being degraded by one of our therapeutic strategies. All right, so with that, I'd like to point out a few people in particular. A lot of the first part of the work was done by Courtney Reichart, also Laura Jennings. The last half, the IHC analysis, was done by Laura Jennings. Courtney is an assistant professor now at WashU, and Laura's an assistant professor at Montana State, so they've gone on to bigger and better things. I'd like to highlight my collaborators. There's one person I accidentally left off the slide who is very instrumental for the immunohistochemistry study. That's Katie Heiser. So very a great facilitator for the work. And with that, I'd love to take any questions if you have them.
Any questions for Matt? Anything online?
I've got one while Sebastian walks up there. No, we're going to wait for him.
Yep.
Okay.
Hi, Matt. Sebastian from Columbia University. Great talk. So I would like to know if you can discuss a little bit more the impact of these EPSs on the ability of the phagocytes to fight the infection, if they are protecting these bacteria or they are doing something else in the CF lung.
Yeah, that's a great question. One thing that's been linked to overproduction of these exopolysaccharides. So if you make a lot of PEL and make a lot of PSL, you form what we call a rugose small colony variant. So it's like a wrinkly colony on solid medium, but in liquid culture, it's highly aggregative. In this, Dan Wozniak's group and others have shown that, phagocytes really can't do their job when you have this, this aggregative mass surrounded by a ton of EPS. So we think there's a link. We also know there's positive selection for this phenotype. So Pseudomonas wants to produce these exopolysaccharides in the CF environment. So, yeah.
Nice talk. This is outside my realm of expertise, so this might be a dumb question, and so forgive me if it is, but the bacteria remain in the shell. They're producing extracellular matrix, and it only gets deposited on the outside. So this is about the tree ring model?
Yeah.
It only gets deposited on the outside, right, of the shell. Do you have any way of measuring the biophysical properties of the outside of the new shell versus the old shell? If it is like a tree, does that dead xylem become hardwood?
Yeah, that's a great question. And to be able to study this, I mean, I have some colleagues who are kind of an expert in this area, like Aaron Gloag at Virginia Tech. But I think being able to spatially study physical structure differences in those regions is really tricky. Maybe Aaron knows of some work that's been done, but yeah, my guess would be is that when you're in the interior, you have a very dense, cell-rich environment that's that's very it doesn't want to move, and you have this ring that is sort of newly deposited EPS that isn't quite as structurally sound as the rest of the aggregate, but
Thanks.
It's hand waving. Yeah.
My question is actually quite similar, and sorry if I might have asked you this before, but do the cells on the perimeter primarily produce EPS? And if not, how do you envision that the new EPS gets where it is?
That's a great question. So we have data that, matrix is linked to cells that have high cyclic di-GMP. So cells want to make PS. And we're doing work in the lab, postdoc of mine, Shuwei Zhang, has shown that there's a subpopulation that's cyclic di-GMP, that's high cyclic di-GMP, sort of co-localizes with the shell. And then you have this interior subpopulation that's actually, that shows high cyclic AMP signaling, which I haven't talked about at all in the talk, but evidence of different types of signaling going on in different regions of the biofilm. So we think it's being made at the periphery. It's not being trucked from the interior cells.
And similarly, when you mentioned that you think it's being recycled, how do you envision that?
Yeah, so, Pseudomonas does have ways to turn these exopolysaccharides over. So there are hydrolases. One thing that would be great, to try and figure out is if hydrolase activity can be linked to, decreased matrix signal, which you see in the interior. And presumably, yes, but we haven't done those experiments. That's what I would guess we'd find, but yeah.
Cool. Great. Thank you, Matt, for a great talk. All right, I'm excited to introduce our next speaker. Can make her way down from the question microphone to the other microphone. And this is Dominique Lamoli. I'm happy to welcome from the University, or Indiana University, Department of Biology. So we've asked Nikki to come down to be our expert on polymicrobial interactions today. And so her lab does some really cool work looking at cellular mechanisms that drive interactions between different species of microbes. So this is where we're going to start to marry the separate Pseudomonas and staph interactions that we've heard about so far today. And Nikki's lab is really interested in identifying ways that we can modulate those types of polymicrobial community behaviors so we can help to improve patient outcomes. So I'm excited to hear from Nikki today about her polymicrobial mechanisms of bacterial persistence in CF disease.
Okay, thanks. Okay. Well, thank you so much for the introduction and for the opportunity to talk a little bit about my lab's interest in polymicrobial interactions in CF. And I have nothing to disclose, but I do like to start with the acknowledgement, so I make sure that I don't forget anyone. The work that I'll talk about today is really takes a village. Almost all of the former graduate students in my lab had some hand in some of the things that I'll tell you about today. And we also have a lot of really wonderful collaborators. And I've highlighted a couple that will, that contributed to some of the work that I'll tell you about. In particular, Dave Terrell and his graduate student, who I co-mentored, Grace Wang, really contributed to the latter part of the talk that I'll tell you about, and also Wilda Paz and Jen Bomberger and her former graduate student, Allison, had a lot of contributed really importantly to the work. And so thinking about polymicrobial infections in CF, I really love to look back to some of the very first reports from Dorothy Anderson when she described cystic fibrosis. And so what she did with these patients is she took autopsies and took lung samples and just homogenized them and plated for different organisms. And what you can see is that there are a lot of the main players that we think about today Staph aureus, pneumococcus, and if you go and look down here at Bacillus pyocyaneus, it's actually Pseudomonas aeruginosa. And my lab is really interested in co-infections between Pseudomonas and Staph. And what you can see is that from some of the very earliest reports, Dorothy was reporting co-infection with Pseudomonas and Staph. And she clearly, in this like predominant organism column and the also present organism column, she was really thinking about polymicrobial infections in these patients. And so if we fast forward to this is the 2019 report from the CF Foundation where they reported these co-infections, you can see that co-infection with Pseudomonas aeruginosa and Staph aureus is really common in these patients. And there are a variety of other co-infecting organisms that you can see here that have been described and have a lot of really important outcomes for patients. But this prevalence of Pseudomonas aeruginosa and Staph aureus co-infections is something that has really been an interest of my lab and various other labs because of how prevalent this is. And one of the first questions that we and many others have had is, okay, well, we see that co-infection is really common, but does this have any outcomes for patients? And indeed it does. So we see that co-infection is associated with a variety of poor outcomes for patients. So they have more pulmonary exacerbations that bring them into the hospital, decreased lung function, increased inflammation, and shortened lifespans. Also, when we and others co-culture Pseudomonas and Staph together in the lab, we see that there are a variety of reciprocal effects that these organisms have on each other. So you can see increased antibiotic tolerance, enhanced virulence, so a variety of different things that we see that happen in the lab that are quite likely to have an influence on outcomes for patients, but making this connection between these effects that Pseudomonas and Staph have on each other when we grow them in the lab, and whether or not that has direct effects on the outcomes for patients is a really tricky question to answer that we still don't have an answer to, but I'll tell you a little bit about the various ways that my lab has been interested in trying to address this question of whether or not actual interactions between Pseudomonas and Staph could affect patients, and if so, what might we do about that? So one of the ways that my lab has been interested in looking at this is first just asking whether or not Pseudomonas and Staph are even found spatially near each other during infection in order for us to envision that perhaps they could affect each other's behaviors during infection. And so the way that we've done this has been by using the method developed by Will DePaz to visualize in situ these bacterial communities. And so just in brief, what we've done is we take explanted lungs from patients who have undergone lung transplants. We fix the tissue, embed it in a polyacrylamide matrix, to maintain the spatial organization of the microbial communities as well as the host environment as best we can. We label the bacteria and then we image. And so here's just one example of what we found. And so what you're looking at here is a lectin labeling for mucus in red. And so this gives us the kind of the outline of we're looking down through a bronchial. And then DAPI labeling the host cells here. And if you can kind of see this orange aggregates, these aggregates are bacteria. And if we zoom in, what you can see is that we've labeled Pseudomonas with 16S ribosomal RNA probes in green and Staph in this orange color. And so what you can see is that Pseudomonas and Staph are forming aggregates that in the lung and they're proximal to the airway epithelium. Quite likely, we haven't labeled like Matt did for polysaccharides, and so probably some of the they're probably embedded in a self-produced polysaccharide matrix that we just haven't labeled. And there's also probably something that we haven't labeled between the aggregate and the epithelium. They're not just like hanging out in space the way that it looks here. But what you can see is that Pseudomonas and Staph are making aggregates together. And this is actually reminiscent of what we see if we grow Pseudomonas and Staph together on monolayers of CF-derived bronchial epithelial cells. And so here, Pseudomonas and Staph are constitutively expressing either red or green fluorescent proteins. And you can see that, again, they're making these aggregates that we see together. So what we're seeing, at least in this in vitro model, is similar to what we're seeing in the lungs of patients. And what we found actually is that we saw these co-aggregates at frequencies that were higher than what we really expected to see. And so we're curious if Pseudomonas, if there's something that actually brings Pseudomonas and Staph together, or if they just happen to like the same thing in the same place at the same time. And so here we asked if we could visualize the very earliest events of these mixed aggregate formation. And so here we took a very different approach. This is a much more in vitro approach, but we wanted to be able to visualize Pseudomonas and Staph on the single cell level. So here what you're looking at is Pseudomonas and Staph that we've inoculated between an aureus and a cover slip, an aureus pad and a cover slip, so we can maintain them in one plane and visualize them over time. This movie, they initially start as single cells, but I'm showing you beginning about 4 hours into the co-culture. Pseudomonas is here as a rod, and it's motile on a surface by type 4 pilus motility, which I'll tell you more about later. And then Staph is non-motile and just grows in these clusters. And so what we saw when we start the movie is that hopefully, there we go. Pseudomonas senses the presence of Staph and actually increases its motility, moves as single cells, and moves towards Staph. And you'll see more Pseudomonas cells that come from outside the field of view, and they start to surround the Staph colonies and will actually then eventually invade into the Staph colonies. And so my lab has been really interested in all of the various aspects of this behavior, and I won't have time to tell you about this today, but if anyone's really interested in more of our molecular studies of how Pseudomonas senses Staph, I'd love to talk with you about that more. But if we label Pseudomonas and Staph again, or with different fluorescent proteins, you can see that Pseudomonas labeled in cyan will invade into the Staph colony. And so we think that this is possible, that this attraction of Pseudomonas towards Staph is one way that they form some of these mixed aggregates. And so, as I mentioned, this motility on a surface is driven by the type 4 pilus. So this is a hair-like projection where they extend from the one pole and attach to the surface, retract and pull itself forward like a grappling hook. And so we've done a variety of different controls to show that this movement towards Staph is mediated by the type 4 pilus. But again, what I showed you is very in vitro, and so we wondered whether or not Pseudomonas would be attracted to Staph under conditions that mimic CF airway secretions better. And so we grew Pseudomonas and Staph biofilms in synthetic CF sputum medium. And so here what we did was first we pre-grew Staph biofilms in this medium, and then we added Pseudomonas cells. And so when we added the Pseudomonas cells, what we found is that they were indeed attracted to the Staph biofilms, and we could even see them integrating into the biofilms and then lifting like the whole biofilm up. And if we used a mutant in the type 4 pilus, we saw that they just kind of settled down onto the Staph biofilms, and we didn't see the same kind of like integration and lifting of the Staph biofilm. So more evidence that Pseudomonas could be attracted to Staph in various models that we've looked at. And so we wanted to know what is it that Pseudomonas is attracted to. And so it looked like Pseudomonas was attracted to Staph, something secreted by Staph, and we wanted to be able to set up an assay where we could really test that. So now what you're looking at is bird's eye view down onto an agar, to just an agar plate. We first set up a concentration gradient of either growth medium or secreted factors from Staph, just Staph supernatant. We then spot Pseudomonas, which is the bright circle labeled in blue, and then this haze outward is the type for pilus motility. If we set up a gradient of staph supernatant, you can see that it's actually Pseudomonas will move directionally towards that staph supernatant, suggesting that it is indeed attracted to just these secreted factors. And if, again, we use a type 4 pilus mutant, we don't see any movement at all. And we can measure the direction towards, the distance that Pseudomonas moves towards versus away, and we just create a directional motility ratio showing that Pseudomonas will move directionally towards staph supernatant. And so we use this to ask what secreted factors from staph Pseudomonas might be attracted to. And just to jump over about a year's worth of genetic and biochemical data, we found that Pseudomonas is attracted to these peptides made by staph called phenosoluble modulants that Barbara mentioned earlier. And so these are these 20 to 40 amino acid long peptides. And if we create staph mutants that don't make these, and we test and we set up a concentration gradient, we don't see any attraction. And so these are really interesting because they're not really traditional attractants for Pseudomonas, but they're highly abundant peptides that are made by staph. They have this alpha helical amphipathic structure, and interestingly, they're chemoattracted to neutrophils. They're cytotoxic towards host cells, but we actually don't see toxicity towards Pseudomonas. So we've been really interested in how is it that Pseudomonas might be attracted to these peptides, and how can we get at how that might happen? And so to address this question, we wanted to ask, what is the response of Pseudomonas immediately to sensing the peptides? And so in order to do this, we took advantage of the precise temporal resolution that's afforded by a method called BONCAT. It's bioorthogonal non-canonical amino acid tagging. And so what we did was you can add non-canonical amino acids that have a bioorthogonal tag. Those amino acids are incorporated into newly synthesized peptides. So any new protein has this amino acid with a tag in it, and then you can use that tag to pull out the proteins and then mass spec. Or you could fluorescently label them. In this case, we mass spec them. In order to get the time resolution that we wanted, and also only either pull out Pseudomonas proteins or staph proteins, we can express a tRNA synthetase in Pseudomonas that selectively incorporates those proteins. So now we're only getting the tag in Pseudomonas proteins if we express this tRNA synthetase in it. And that's worked really well for us. The overall protocol of what we did was we added either the PSMs or staph supernatant. We pulsed them in just for 30 minutes to Pseudomonas cells that have this. And then we add in the non-canonical amino acids. Pseudomonas will incorporate this. And so now we can see any newly synthesized proteins that Pseudomonas makes immediately upon sensing staph or the peptides. And then we use click chemistry, pull out the proteins, and mass spec them. And so here's an overview of what we saw. So we saw a variety of proteins that were either increased or decreased upon the staph or PSMs. And so the first two things were great because this was chemotaxis and motility and cyclic diGMP signaling. These were all things that we've seen in our other studies. So that was really nice confirmation that this was similar. But some of the new things that we found were that Pseudomonas actually increases type 6 secretion, which is an interspecies competition mechanism, and pyoverdin, which is a siderophore that binds to iron. And so we were starting to get this picture that Pseudomonas was actually upregulating things that it can use for competition, even just by sensing the peptide alone. So no, without even any other bacteria around, just sensing the signal allowed for it to act as though it were sensing competition. And so to focus in on the type 6 secretion, this again is a needle-like syringe that will, upon either contact with either another gram-negative organism or with eukaryotic cells, Pseudomonas can inject toxic effectors into the cells. And so we were really interested to see whether or not this was actually being activated. So we saw the proteins being increased, but do we actually see this? We need to have firing of the syringe in order for it to have any kind of effect. So we wondered whether or not we saw actual firing. And so to address this question, we labeled an ATPase called ClpV1 that when the type 6 secretion fires, then this ATPase is recruited to the base to recycle the components. And so if you look here, when we see puncta formation, that means that the secretion system was fired, and now you have recycling. And so at a baseline level, we saw like a few puncta. But if we added the PSM peptides, then we saw a substantial increase in this puncta formation, suggesting that indeed we were seeing an increase in actual firing of the type 6 secretion, and we can quantify this by looking, quantify increased puncta per cellular area. And so we were really curious to know whether or not, what were the consequences of this? And so this is bringing us back to this overarching question that we had, which is, are there interactions between Pseudomonas and staph that might lead to virulence in patients? And so we wondered, does this increase firing actually lead to toxicity with airway cells? And these experiments were performed in the Bomberger lab. And so what we did was they looked at cytotoxicity of Pseudomonas towards CF-derived bronchial epithelial cells. And so you can see that, as expected, there's an increase in toxicity when you add wild-type Pseudomonas to the cells. And so then the question was, does this increase upon pre-treating Pseudomonas with the PSMs? And indeed it does. So we see an increase in toxicity towards epithelial cells when adding the PSMs to Pseudomonas. And then the next question is that dependent upon type 6 secretion? And so if we use a mutant that doesn't produce type 6 secretion of all 3 islands, then we actually saw that increase in response to the PSMs is alleviated, suggesting that indeed adding that Pseudomonas can respond to PSMs by increasing type 6 secretion, and that this causes increased toxicity towards the airway epithelium. And so again, bringing this kind of back into an entire model, the way that we envision this is that Staph aureus is secreting these PSM peptides, and Pseudomonas can sense them and is attracted towards it. And instead of moving away, as any sensible organism would do, it actually moves towards towards this. It senses competition and moves towards it. And we think that it can do that because as it does, it's arming itself along the way. It's increasing type 6 secretion. It's increasing the production of siderophores, which can recruit iron and steal that away from staph. And so we think that this is part of how it's able to actually move towards other organisms. And then meanwhile, there's collateral damage to the host airway epithelium as it's increasing these competitive mechanisms that it uses to compete with other organisms are also toxic to the host environment. And so if we have lots of questions to follow up on this, including how Pseudomonas actually regulates activation of type 6 secretion. If there are other signals that can activate this, and we have some preliminary work suggesting that indeed there are a variety of other signals, and what are the consequences of this competition sensing to the regional microbiome? And finally, again, bringing this back to this idea of whether or not competition sensing might actually contribute to poor outcomes during infection. We're continuing to do this spatial imaging, and we can see areas where Pseudomonas and staph seem to be invading into and causing actual direct damage to the airway epithelium. And so we're doing some work of trying to figure out whether or not we see more damage when Pseudomonas and Staph are together. Does this depend, again, on these in vitro mechanisms that we've been looking at? And finally, the ultimate goal is to understand whether or not competition sensing could be inhibited and whether or not this might actually improve outcomes during co-infection. So again, just love to acknowledge the folks in my lab and the funding. And I would love for anyone to scan the QR code if you're interested in ways that we're working on making our lab more sustainable. And happy to take questions.
Please remember to state your name and your affiliation before your question.
So my name is Jacob Harper. I'm from Sniper Bio. Thank you very much for a very fascinating talk. I was curious about it. I mean, it's very clear that the Pseudomonas are attracted to staph. And after they find each other, do you find then a synergistic or an antagonistic interaction between the two bacteria? And as a follow-up question to that, if you have any kind of hypothesis, what would happen if you would specifically take out one of those bacterial populations? What would happen to the other one and the interaction with the lung?
Yeah, so the answer to the first question is a little complex. So we see a variety of different effects depending on the nutrient conditions as well as the strains. So our laboratory strains of Pseudomonas are very toxic towards staph. And so in a lot of our in vitro studies, Pseudomonas makes a variety of different staph antimicrobials. And so generally, Pseudomonas is pretty toxic. However, a lot of the clinical isolates that we get from patients with CF, especially those that come from co-infected patients, are less toxic. So one aspect of why there's such high levels of co-infection, we think, is because there is a reduction of some of those Pseudomonas antimicrobials. We do see that those isolates are still attracted to staph. So while there is some killing as response, it's not necessary for the response either. And for the second question it was what effect we would see if you eliminated one or the other.
Yeah, so say you could actually kill specifically Pseudomonas, what would then happen to the staph?
Yeah, that's a great question. I can only, I think, so we did, in collaboration with Tony Fisher at the University of Iowa, we did a study where we followed patients longitudinally with the idea that, if we look at these graphs from the CF Foundation, when you look at populations as a whole, it looks like there's this clear inverse relationship between the increase in Pseudomonas and a decrease in staph. But if we followed patients, we didn't necessarily see that. And so whether or not you would see, I think that there's some regional antagonism, but what we saw is that there's such a high abundance of Pseudomonas and staph in the airways that there's just so many things that are going on. So even if there's some regional competition, I think there are areas where there's not competition. And so I don't think it's sufficient. Yeah.
Thank you.
Hi, Nikki. Thanks for a lovely talk. I'm Valerie. I'm from Sick Kids, and I think we've met before. So I had a postdoc who did an observational study, I think you know Amanda, where we collected sputums from co-infected patients, about 50 different patients, and used the same methodology. Mm-hmm. And we almost never saw co-localization. That Pseudomonas and staph existed very distinctly as two different populations forming aggregates. Yeah. And despite I thought we would see this co-localization, but we hardly ever did. It was incredibly rare. So I wonder exactly, as you were previously mentioning,
Yeah.
if the stage of infection and the strains involved are really important to know whether they're actually physically going to interact or not.
Yeah.
In our population, we didn't see it.
Yeah.
In your image that you were showing, these were explanted lungs, right? So clearly very end-stage lung
Yeah.
disease and maybe quite a unique situation, right? That they're
Yeah.
going for transplant. So this
Yeah.
kind of invasion that you see, I don't know if that's going to be true for all our co-infected
Yeah.
patients. So
Yeah.
I don't really have a question, but other than to
Right,
say
I mean,
we just
I-
very different. We never saw
Yeah, absolutely.
coagulates
I
like
do think that there are going to be many situations where there are, depends on the stage of the patient. We have collected sputum samples and we do see some co-localization. We see co-localization and we see not, right? Like we see both.
right.
We certainly see more not, we see them separate more often.
Yeah.
And because of your work and others, that is what we expected to see more often. And so that's why we were surprised by the frequency that we saw. I definitely think that it has a lot to do with the strains.
Yeah.
And so one of the things that we have noticed, and we've been really interested to do more of, especially in, like it's similar to what Matt has done, is that we also see really clear differences in our Pseudomonas aggregates of whether or not they incorporate different host factors into the aggregates as well. And so there are some that really incorporate eDNA and mucins, for example, into the aggregate, and some that exclude them, where it almost looks like there's like a separation, a distinct separation. And so based on some of our work and others of showing that like mucoid Pseudomonas is less antagonistic towards staph, it seems like we hypothesize that perhaps there's a polysaccharide phenotype that allows for, the charge of the polysaccharide is going to, influence its interaction with different host components. And then I think the antagonism with other organisms as well. So we see a huge heterogeneity in the way that the aggregates look and whether or not they incorporate host components and also other organisms. And we also see that same thing with organisms that are not staph.
All right. Thank you, Nikki. Thanks.
So we're coming up to time, so short questions would be great.
Okay. Katie Heiser from National Jewish Health. The type 6 secretion is really fascinating, and you introduced it as it can affect other gram-negatives and host cells, but staph is neither of those. So can you comment on, do you know if it's affecting staph at all? And then you did mention that the lab strains are more toxic than the clinical ones. Have you looked at clinical strains to see if they also upregulate type 6 in response?
Yeah, those also a complicated answer. So we we have seen conditions where we do see type 6 dependent inhibition of staph. We think that the mechanism is quite different than it is with gram-negative organisms, which is part of why I glossed over it. And so that's something that we're working on, and hopefully we'll have more of an answer for.
Great talk, Tanya Wong from Rutgers University. Normally, type 6 secretion systems, if they are contact dependent, like they would normally subvert host cell responses, like actin cytoskeleton, but here you're showing cell death. So that's really cool, but have you tried to characterize the type of cell death? Is it to release metabolites that would then feed staph and Pseudomonas altogether? And also, have you looked at contact-independent type 6, where they could be sensing the environment and responding to that?
We haven't done the first question, but I love that question, and we'd love to look into that further. We do think, so the PSM activation is contact independent. And so the activation of type 6 indeed is, and how that works is also something that we're interested in. Yeah, thanks.
Thanks, Nikki. All right, and now I'm happy to introduce our last speaker for this session. So we've heard a lot about some of our traditional CF pathogens that we think about a lot, Staph and Pseudomonas. And then today I'm excited to have Paul Planet from CHOP and UPenn close out with a little bit more about some of the bugs we don't talk about maybe as much, but should think about more. And so Paul is an assistant professor of pediatrics and infectious disease. He uses a lot of cool sequencing approaches to think about kind of mixing sequencing bioinformatics with traditional microbiology to think about evolution and ecology of some of these less studied organisms. So we'll be happy to hear about Paul's perspective on these today.
Great. Thank you, Megan and Bob, for the chance to talk about anaerobes. Nobody lets me talk about anaerobes usually, so this is a nice opportunity for me. I spent so much time thinking about anaerobes and putting this talk together that I jettisoned the idea of emerging cystic fibrosis pathogens. I'll mention maybe some of them because I am interested in things like NTM. But I'm really going to focus on anaerobes here because I think they're an enigma. So, am I too loud? Should I back up? It sounds really loud. Okay, so my disclosures are that I'm funded by the CFF to study an anaerobe, Prevotella melanogenica. I also consult for them, but it's in my best interest to make you think that anaerobes are really important. So we'll start with a clinical question. So a 15-year-old girl with CF presents with a CF exacerbation that you think requires hospitalization for IV antibiotics. She usually grows mucoid Pseudomonas. Because of a lab error, her sputum is accidentally cultured anaerobically, and the culture is reported as heavy growth of a beta-hemolytic Prevotella melanogenica. And what do you recommend? So I actually asked this maybe 2 or 3 years ago at the NACFC, and I had people vote on it. And the votes were all over the place. But I'm just sort of pointing out to you that in each one of these regimens, B ignores the anaerobes, whereas all of the other ones address the possibility that the anaerobes are involved in this disease. So really, this comes down to the question of, do you think that anaerobes are bad in some way or good? So this is the outline of the talk. I'm going to talk about what might make you think that anaerobes are pathogens, what might make you think that they're sort of facilitators of disease, and then also visit the possibility that they might be good guys. And whenever I think about this question, I come back to this paper, which is quite old now, where there was a head-to-head comparison between meropenem and tobramycin and ceftazidime and tobramycin. And in that question, the ceftazidime and tobramycin was the one that ignores the anaerobes, right? So the meropenem is a very good anaerobic drug. And in this study, they looked at time to get, it's sort of a weird y-axis, but basically it's time to get better, right? And the ones who got meropenem got better faster. They all got, eventually they all got to the same place, but the ones who got meropenem got better faster, suggesting that maybe if you're interested in things like length of stay in the hospital or the amount of days that you're not feeling well, that you might want to address an anaerobic component. There was a subsequent study by the same group that went back and looked at the same data again and kind of found a less significant result. So you can see this, that the meropenem in sort of overlying this in getting better over time compared to ceftaz-tobra. But there was this suggestion that still might be the case. And then this is a slide that I stole from a collaborator who I'll mention later. But this is work outside of CF where they looked at the presence of anaerobes in patients who had very severe pneumonia, and the ones that had very severe pneumonia and had anaerobes seemed to do better. So what are the anaerobes that we think about in the CF lung? Prevotella really is the top of the list, and I'll show you some more data about that in a second. Veillonella, Fusobacterium, Porphyromonas, all of these are things that live in the oral cavity, some of them just free in the oral cavity, some of them in the periodontal pocket. I'd also add Rothia, a gram-positive anaerobe, to that list. But they're really there in the CF lung. So from the perspective of, are there anaerobes in the lung, not just in the mouth? The Prevotella species can be found at really high CFUs per gram in the sputum and BAL. Quantitative PCR supports that there are really high levels of anaerobes, at least to the levels of traditional pathogens. There's also been culture-enriched profiling that says that they're about equivalent levels. And then there's the idea that, of course, because there's so much mucus plugging in the CF lung, it's kind of an ideal place for anaerobes to live. This is really old data from my lab, where this is back when we were using 454 sequencing for 16S, but it holds up over many different studies, not just from my lab, where I've coded the anaerobes are blue and the sort of traditional CF pathogens are red, and when you look at it in the number of samples that have one of these anaerobes, Prevotella, Veillonella, Fusobacterium, all those anaerobes really top the list, and the traditional pathogens are farther down. And it doesn't really matter which way you cut it. The number of patients, the number of samples, the number of reads, those anaerobes are always rise to the top. And this is much more recent data, which all of you can access if you want to go to Microbiome DB. This is a pediatric study that we did in my lab, where we looked not using 454, using Illumina, and you can see that 96% of the samples that we had from this study, this is all in children, had Prevotella in those samples. So this is a completely ubiquitous organism in CF. So what are the possible roles? So one is that these are active pathogens. Do they make toxins, proteases? Is there some way that they can stimulate the immune system? Another possibility is that they're facilitators of disease. Maybe they make something that the sort of traditional pathogens use to cause worse disease. Or maybe they protect the traditional pathogens. Or maybe they're mitigators of disease. Maybe they dampen the inflammatory response. Or maybe they can kill the pathogens. There's not much evidence for that from the anaerobes. Or maybe they're just innocent bystanders, which I won't talk about. Okay, so what's the data that suggests that they're active pathogens? So there's Prevotella intermedia, which actually has a complement inhibition phenotype where it can degrade complement. There is a hemolysin that we've been interested in my lab for a long time in P. melaninogenica, and actually this hemolysin is found throughout the Prevotella. And then there have been studies where they show direct toxicity to neutrophils in culture. I'm just going to show you an example of the hemolysin from melaninogenica. This is two lab strains on the top. One of them is a very strong hemolysin producer. The other one is the type strain, which is a somewhat okay hemolysin producer. And then the bottom is a bunch of strains that we collected from CF patients with varying levels of hemolysin. But they're all making some level of hemolysin, which suggests that if they ever run into a red blood cell, they could get some heme from it. So we wanted to, and this actually, with this particular hemolysin was actually discovered quite a long time ago in a 1997 paper where they identified it as this Phi-A gene. We've gone on to characterize this a little bit more, and there's also an upstream gene that seems to regulate that Phi-A gene. And then we wanted to know if a lot of hemolysins double as toxins, right? They double as toxins that can kill other cells. And so this is work that's done just with some of those P-mel strains and compared to PBS, and also in sync with their ability to make the hemolysin. They are very good at both lifting up monolayers of epithelial cells and also killing them. I'm not going to show you the data here, but if you heterologously express these the toxin in E. coli, you can actually get exactly the same phenotype when you put the E. coli on cells. Okay, so the next possibility, I shouldn't clap near here, the next possibility is that they facilitate disease, right? So, and really there's been a lot of work around this, and the thing that anaerobes are sort of famous for doing is degrading mucins, right? So they can take these really complex carbohydrates and break them down into things that other organisms can eat, and perhaps very importantly, short-chain fatty acids. Those short-chain fatty acids have been shown in the lung to correlate with bringing in neutrophils, with pro-inflammatory cytokines like IL-8. And then in various other studies, there has been work to look at the fermentation products of anaerobes and how they all support growth of other organisms. And then there's also this idea that one of the first steps in an exacerbation is the formation of this attack community where the anaerobes come in, they ferment the mucus, they get things ready for the pathogens that then come in and do the damage. And then there's this other cool paper where they looked at Prevotella species, and they saw that in 75% of Prevotella species, they're making extended spectrum beta-lactamases, which would be the kinds of beta-lactamases which would destroy critical drugs like ceftazidime. And this is in vitro co-culture where they've put Pseudomonas and Prevotella together, one of these Prevotella that is expressing an ESBL. And just the fact that they're growing next to each other, and because an ESBL is an enzyme, so it chews through lots of beta-lactams at once, the Prevotella is actually able to prevent killing from ceftazidime. Okay, so one thing that these two ideas might suggest is that Prevotella is kind of protective of the microbiome as it stands. And this is work also from my lab. It's an alluvial plot where we're plotting consecutive microbiomes from sputum samples, so the sputum and also oropharyngeal swabs and some BAL. And we're just saying, what is the dominant species from one time point to the next time point? And you can see that for certain species like Streptococcus, you have really high stability of the dominant taxon from one time point to another. Prevotella also seems to be pretty stable. It's not going to many other places, whereas if you're down in the Pseudomonas land, you could, your next dominant taxon could be many, many different things, right? So we wanted to sort of quantify this because I think this is very beautiful, but it's hard to quantify, right? So we used a Markov model, and we looked at, just given the relative abundances of these organisms, what's the chance, the expected chance that the next one you're going to see that dominant one. And it turns out that for both Streptococcus and for Prevotella, the expected chance is lower than the observed chance. So what that's saying is that there's some sort of stabilizing quality of having this as the dominant organism. So maybe coming back to the fact that Prevotella might be feeding other organisms and keeping the microbiome intact. The other important thing here is that when you do change a dominant organism in CF, you're very often changing your clinical status. And we just use this definition of baseline, exacerbation, treatment, and recovery that suggests that you're much more likely to change your clinical state go from baseline to exacerbation, for instance, when you switch your dominant organism. So the more stable communities are more likely to keep you in this whatever state you are. There are problems with that, but we can talk about that later. Okay, and then the last point is maybe these anaerobes are actually good. This brings in a lot of data from that anaerobes tend to be associated with higher diversity. And while higher diversity is not necessarily good, it tends to be better and also is associated with better lung function. I'm going to show you data that's going to show you that anaerobes are often anti-correlated with the sort of traditional pathogens. And then there's also data that I'm going to show you that suggests that there might be a real basis for this in the lung. Okay, so this is data from Marianne Muhlebach that suggested that when anaerobes are present, your lung function tends to be better in the setting of having Pseudomonas. It wasn't the best group, but anaerobes seem to be associated with better lung function. And this is also data from my group looking at both diversity and lung function when your community is dominated with different organisms. So when you're dominated with Prevotella, your diversity is high and your FEV1 tends to be higher. As you get into the more gram-negative standard pathogens, you see that both the diversity and the lung function fall off. And you also see really strong structuring of the community based on which anaerobes you have. So the two major components in this principal component analysis are Prevotella on the bottom and Rothia on the other side. And you can see that there is a correlation there with lung disease. This slide didn't completely translate to the PC, but what we're looking at here is the relative abundance of Pseudomonas and the relative abundance of staph, and you can see that Prevotella, Rothia are really associated with, are anti-correlated with those two top pathogens. And Sarah Clark presented some of this work yesterday morning. It's really compelling and interesting work showing that Prevotella working at the epithelial barrier, potentially by stimulating a TLR2 response, does two things. One of those things is it increases IL-8 and IL-6, and that it might also be through that short-chain fatty acids, which then call out to the neutrophils. The neutrophils come and eat the staph. And then the other thing that's going on through a mechanism that we really don't understand what's going on yet, but is that for whatever reason, staph is less able to adhere to that epithelium. In the CFTR context, however, Prevotella really isn't able to do that. And so from the standpoint of anaerobes maybe mitigating disease, CF lungs might not be being as mitigated as a normal lung. And that I think that bears out in the fact that there's so much Prevotella in the lung to begin with. There's a lot of hope and some hope from Sarah's work that on modulators you actually will see a return to the wild type side of things from both of those responses. Okay, and Katie's in front of me, so I had to show Katie's work. What happens to Prevotella when you give correctors? The answer is it stays pretty much the same. And this is even more borne out in more recent work with ETI, where you see decreases in many of the other standard pathogens, and the anaerobes stay pretty much the same. I don't understand this. I really don't understand this. Why is it that your lungs know what's an anaerobe and what's a traditional CF pathogen? That doesn't make sense to me. So there's got to be some sort of something that anaerobes are doing that is either not affected by the regaining CFTR function, or there's I just don't understand it. Please tell me if you understand these data. Okay, so I'm going to wrap up a little early, it looks like. So the anaerobes. So I actually really think that thinking about anaerobes as a class of organisms is kind of stupid, right? They are a huge group of organisms. They come from different bacterial phyla. I've mentioned multiple phyla just standing up here. And it's probably true that they are pathogens, facilitators, and ameliorators. And that probably goes down maybe even to the strain level, certainly to the species level. And Sarah has some really interesting work comparing different species of Prevotella, and different species of Prevotella don't have that same impact on mitigating disease. So in CF, that mitigator role might be less important, and that might explain some of the overgrowth of Prevotella in that species, in that situation. And then anaerobes they don't change much when in modulator treatment. So we're now going into a place where we may have very high levels of anaerobes in the lung, even when people are being treated, and that could have all kinds of effects. They do have things like LPS that can stimulate an inflammatory response. And my acknowledgements here, as we were going along, a lot of this work was done by Priyoti Sarwar. A lot of that is unpublished data, and we're working on trying to get it out right now. I've mentioned her a lot already. Sarah Clark and I have been really thinking about this ability of Prevotella to mitigate inflammation and disease. Thank you.
We'll just wait for Katie to get up there and ask her question.
Hi, Paul. So I have a question, and then I have a possible explanation, which I feel like is extremely controversial and could cause like a bunch of comments. So question.
I hope you're safe.
So question. You did say that anaerobes is too big of a category.
Yeah.
But I mean, even within that, and I think I've definitely talked to Sarah Clark about this. I mean, there's Prevotella and there's Prevotella, right? It's not like one species, and then probably even within a species, there's different isolates. I've been thinking about this a lot with Pseudomonas, and when we do clinical trials, it's like, do you have Pseudomonas? Do you not have Pseudomonas? But there's different Pseudomonas. And so do you think that that is getting lost in when we do these observational clinical, categorizations, that if you just say Prevotella, it's too big of a bucket?
Yeah, I think definitely. And some of the work that I didn't mention that Priyoti has done is she just took Prevotella melanogenica and asked if we apply sort of normal taxonomic criteria to it, is it one species or not? And it's not. It's like it's like 5 or 6 species, and with very different genomes. So I think we are losing a lot. And I think it's because the taxonomy isn't really well developed. You think of something like Staph aureus versus Staph epi, or Pseudomonas aeruginosa versus Pseudomonas fluorescens, right? Huge difference, right? You would never treat those things as Pseudomonas, right, or Staphylococcus. But we have, for the entire genus of Prevotella, we say Prevotella. So,
and then the comment about the interpreting the data from the modulator trials. Some of us might think that those anaerobes are in the mouth and throat and into the lungs, and maybe increased mucociliary clearance doesn't affect that. But if you actually live in the lung and you have increased mucociliary clearance, then your total and relative abundance will go down. That's just one interpretation of the data. So
Yeah, I agree. I think it's sort of hard for me to swallow that pill when I present a bunch of evidence about how many anaerobes are down there. But there maybe that there is something to that. Not so controversial.
Matthew Bigart, Kansas University Medical Center. As we start using ETI on younger and younger patients, do you think they'll develop anaerobes to that same high level you're seeing?
Yeah, I mean, the data, it seems to me like let me go back to our data from the pediatric study. They really actually had really high levels of anaerobes, and if you plot it by age, the anaerobes tend to actually be earlier and earlier. Now, maybe having ETI early would decrease that. But some people also have described this sort of as a succession of, going from first to anaerobes, and then they get that community sets up the next stage for the pathogens to come in. So I don't know I don't know the answer to the question, but I think it would be really interesting.
Thank you so much.
Hi, Yasmin, Dartmouth College. I'm going to come up with a slight rebuttal to Katie. And so we take sinus samples endoscopically. We're not taking anything that's coming from the mouth. And I actually see tons of Prevotella in those samples as well from people with cystic fibrosis. And so off the back of that, I do have a question more generally, which is in the lung, I feel that there's much more of an anaerobic environment for these bacteria to live in. So what would you think is the mechanism for them actually surviving in the sinus where we're not getting sputum? There's still mucus and it's not being cleared, but not to the same level. I just wondered if you could comment on co-culture and maybe how they were surviving up there.
Yeah, I mean, I've always been really interested by the idea that there's so many anaerobes that live in our mouths, and yet there's plenty of air in your mouth, right? There are places that tend to have more of them, right? So you get the periodontal pocket where it really is down in there, and you're not getting a ton of oxygen there. I can only sort of guess that it has something to do with the sinuses not being as aerated as the rest of but even in exposed parts of the oral cavity, you get these anaerobes. So.
Thank you.
And one other thing I'll mention is that a lot of these anaerobes are actually pretty good at surviving. Like when we take Prevotella out of the anaerobic chamber and we put it on the bench, we can still do stuff with it before we put it back in.
I have the same experience working with it anaerobically.
Yeah, exactly.
Hi, Paul. It's Fabrice. Great talk. I'm always happy to hear stuff about Prevotella. So maybe an interesting question for you. Do we need to care about which type of anaerobes are in the CF lung? I mean, there are some studies showing that people with CF with either strep or Prevotella have good clinical outcomes. I would like to get your thoughts on that.
You mean which species?
For instance, you're showing that Prevotella seems to be quite important, in the post-ETI era. But if you were to think about not specifically about Prevotella, but just streptococci, for instance, do you think it really matters? Should we be enriching for Prevotella or streptococci?
Right. I mean, I think this is a really important new direction. If we do know that there are either good or bad effects of different anaerobes in different places, then I don't think that there is in the end of the day that there's going to be a basic quality of all anaerobes that's going to that you can just pick any anaerobe and that's going to be good enough to do the thing you want it to do, push it in the right direction. There does also seem to be a predilection for certain anaerobes, like there are predilection for certain aerobes, like Pseudomonas aeruginosa, like melanogenica shows up over and over and over again in many, many different people, much more so than other Prevotella. So I don't know what that means in terms of which we should favor and which one we shouldn't favor, but I think it probably matters. And like I was sort of alluded to with Sarah Clark's work, different Prevotella interact with the immune system differently.
Hi, Paul. Sebastian from Colombia. So trying to understand for the mechanism that is keeping us awake at night. So I think I would like to make a little bit of justice to the immune system, you know, because we are giving patients CTI and the bacterial burden is going down. So maybe the immune response has something to do with it. As you say, very beautiful describe, major difference between the anaerobes and the other CF pathogens is their ability to produce energy through oxygen. So is it possible that after giving ETI, we are restituting the ability of the immune cells to kill pathogens that use oxygen to produce energy? So we are restituting that mechanism, and that's why the anaerobes are not being affected.
I like that. That's a really interesting idea, that there's something about the actual immune response that specifically kills the aerobes because the anaerobes can deal with it. I like that. Shall I name that hypothesis after you? The Riquelme hypothesis.
Yeah. Well, cool. Thanks.
Okay, thanks, Paul.
Thanks.
All right, I'd like to thank all of our speakers for 4 great talks that really highlighted a lot of the cool observations we've been able to make about CF microbiology over the past probably 20 years plus that we've seen today, and a lot of important questions that we still have to address that are worthy of, I think, attention in the CF field and continued interest. So thanks everybody for attending, and we look forward to seeing you around.